Hey guys, welcome back to our second video for photosynthesis. Today we're going to go over the light-dependent reactions involved in photosynthesis. This is going to be a much more detailed look at photosynthesis than you've seen before if you've taken Gen Bio. So let's jump right into it here. This is your general equation for photosynthesis, and so kind of bear this in mind because we're about to get into a lot of detail, but big picture here. Plants bring in carbon dioxide gas. They also bring in liquid water. They also bring in light energy, and they're going to make sugar, and they're going to make oxygen. So you should know this. Carbon dioxide plus water plus light energy is what plants use to make sugar and oxygen. And of course, you know already that humans rely on the oxygen gas. So real quick, we're going to pause and I'm going to show you a video so you can have a really firm grasp on exactly where we are in a plant for all of the things that we are about to talk about.
Inside a plant leaf, we find layers of photosynthetic cells. These cells contain many chloroplasts. These are the organelles of photosynthesis in plants and many protists. A chloroplast has three membranes. Two outer membranes enclose an enzyme-rich semifluid interior called the stroma. This is where sugars are built. A third membrane, the thylakoid membrane, forms a series of interconnected, flattened sacs that...
Okay, so I'm going to pause this real quick. If you want to sketch this real fast or write these down, do whatever you want to do, but this structure here is very important for you to understand. This is the chloroplast. It's the organelle in the plants. The two outer membranes, it's a double-membraned organelle like so many others. Those are important, but they're not quite as important perhaps as these. This stroma is the term that means the fluid inside of the chloroplast. And all of these little things that look like a stack of pennies, these little stacks of pennies, the individual penny would be a thylakoid disc, and another thylakoid disc, and another thylakoid disc. And so what makes up the surface of that penny, that's called our thylakoid membrane. Now the first reactions that we're going to talk about for the rest of this lecture are all taking place right here on the surface of this little tiny thylakoid membrane. And then later on in other lectures we're going to get to what goes on in the stroma. And so having a good bearing for where we are geographically inside of an organelle will make this task a little less daunting.
Through the stroma. Embedded in the thylakoid membrane are light-harvesting complexes, photosystems, electron transfer chains, and ATP synthases. Together, these membrane components capture light energy and use it to synthesize ATP and NADPH.
All right, so that's where this stops, and that's actually where our discussion begins, is if this whole structure here is one thylakoid disc, and this is the membrane of the thylakoid. Today, you and I are going to focus on just this little tiny area inside one little tiny structure in one little tiny organelle. So it's a lot of detail, and it is kind of a lot to take in. And so let's jump right into it. So our first stage of photosynthesis is called the light-dependent reaction. Light-dependent means that we absolutely require sunlight or light from a light bulb or something to get these reactions going. And we call this non-cyclic, which means this pathway is not a cycle. It has a beginning and an end, and the end products do not cycle back in to the beginning of the reaction. A big picture for light dependent is we're going to take light energy from the sun and we're going to turn it into ATP, which you guys will recognize as the energy currency for a cell basically, and NADPH, which you'll recognize as another energy carrier. Water molecules are going to be split in this process, and when you split a water molecule, you get hydrogens and you get oxygens, and so we will release O2. That's where the O2 gas that you and I rely so heavily upon comes from. And this is going to occur on the thylakoid membrane, like that video I just showed you, just that little membrane of those little pennies, those little thylakoid discs. That's what all our discussion today is just on those membranes. So the thylakoid membrane is a membrane that makes up thylakoids, thylakoid discs, And these membranes have clusters of light harvesting pigments. You might remember that from our last lecture, things like chlorophylls, for sure, the heavy hitters there, that absorb photons of different energies, like we talked about last time, and they convert light energy into chemical energy. And that piece right there is what makes plants just so incredibly important. Light energy into chemical energy. For biological systems, like, for example, you and I, and all the food that we rely on, these guys are very, very important. So, our second stage of our light independent reactions is once we get that energy to the ATP, which is a topic we're going to get into in just a minute. We're still kind of big picture here. We get that energy in ATP and we get NADPH, which is carrying some energy too. And we're going to take those guys out and actually make glucose with a whole other system we're going to talk about next time. And we're going to make other carbohydrates. We can, combine the glucose and turn it into sucrose and then pump it all around the plant from that. And we're going to do that from CO2 and water, which are very initial inputs that we talked about a slide or two ago. And so that piece, which comes a little later, does not happen on the thylakoid membrane. When it comes to making glucose, we're doing that in the stroma, which is why I showed you the video earlier. The stroma is that fluid outside of the thylakoids. That's where sugar is made. And so here's the semi-fluid matrix surrounded by outer membranes, and sugars are built in the stroma. So big picture, if we were in Gen Bio, we could probably just call it right there. We've got light, the light goes to ATP, the ATP makes the glucose in the stroma, and voila, sugars. But this is not Gen Bio, and we're going to get a little closer look at just how incredible all this little machinery is that we're going to be looking at. So here's your chloroplast. This is what you might see in a microscope. You can see a lot of the cell is see-through. Why do you think that might be, bio peoples? Why can I see through all this blue stuff? Hopefully you're thinking that's a giant central vacuole and it's a huge water bag and everything else is pushed to the outside. And if that's what you were thinking, you're absolutely right. Most of a plant is water. Some cells, 90% of the space can just be that central vacuole. And so chloroplasts, nuclei, they're all shoved way out here into the outside. So you take a look at one chloroplast, and he looks like this, which we just saw in the video. And then you take a look at one thylakoid, and you look at how this is a little bit bigger. We're actually taking a look at about 6 thylakoids. And you can see that we have a compartment, which is a hollow space on the inside of those thylakoids. And that's where we're going to be focusing right now. So, light-dependent reaction. The first thing that happens is we convert our light energy into chemical energy, which you guys already knew we were going to do. Photons, so our energy that is coming from the sun, photons are going to boost electrons in these pigments that we already talked about. And when we get all that energy from the sun into electrons, we get those electrons into some higher energy levels. And we're going to use that energy from those electrons down the road. And so we have these light harvesting complexes that you're familiar with. We talked about chlorophylls making light harvesting complexes with magnesium in the middle. And all these electrons that are hitting the leaf are kind of transferred to these light harvesting complexes. I've got a document in your folder for this chapter, and it's got some silly name like useful photosynthesis videos, and I think there's only one video on there. Please take the 4 minutes out of your day to watch that, because that will make these pieces right here so much easier to understand. So, electrons are released from special pairs. So there's a couple special chlorophylls that we want to get this energy to, and those electrons will be released from those special pairs of chlorophyll A in the photosystems. And these electrons can be used in a non-cyclic pathway, which is what we're going to talk about first, or they can sometimes be used in a cyclic pathway. And so we'll see what that looks like. Here's just a zoomed in view of light harvesting complexes, these little tiny green star-shaped fellows all over the surface of a thylakoid disc. This is the surface of a thylakoid membrane. And then you have photosystems all over the place. Photosystem 1, photosystem 2, near these light harvesting complexes. And so that's what the very, very zoomed in view of a thylakoid membrane might look like. Alrighty, so non-cyclic pathway. Our photosystems, there's type 2 and type 1. Please note that they are not named in the order that they occur in this process. They are named in the order they were discovered. And so photosystem 2, it comes first in the process of photosynthesis, and photosystem 1 is used later. It just happens that photosystem 1 was discovered prior to photosystem 2. So they are named a little bit backwards, and they're the ones who contain these special pairs of chlorophyll that we mentioned earlier, and they're going to be the ones who gather these electrons and move them to where they need to be. So when we're losing electrons from photosystem 2, which is our first photosystem, we need to replace them, and we're actually going to replace them by breaking water. Photolysis of water. So lysis is to break or to burst. Photo are like photons, these little packets of energy from the sun. And so when the energy from the sun breaks the water, it's going to give up some electrons, and we can replace those in photosystem 2. So that is right there, what I just said. And electrons that are lost from a photosystem are going to enter an electron transfer chain, where we're going to make sure we get the most we can out of those electrons. And that is all occurring on our thylakoid membrane. So very, very busy place, the thylakoid membrane. All right, so in our electron transport chain, our electron energy is going to be used to build up a hydrogen gradient. And we're going to get a hydrogen gradient across that membrane. And hydrogen is super important. You might remember this from GenBio. Hydrogen is the fuel that moves ATP synthase, which is an incredibly important and pretty decent sized protein that is going to grab ADP, adenosine diphosphate, and then inorganic phosphate, and it will add that third phosphate group to ADP and turn it into a really high energy molecule. And so ATP is going to be formed, which is very good news for us, and it's actually going to be formed in the stroma. You'll see that in a video I'm going to show you in just a second, where that ATP kind of ends up. And it ends up in the stroma for a very important reason. We don't want the ATP inside of the thylakoid because it's no good to us there. We want the ATP in the stroma because the stroma is where we need that ATP to build glucose. And so that's what we're going to use ATP for. And we are using hydrogen ions to make that ATP. So here's our non-cyclic pathway. It is wordy. Here we go. Electrons from the first electron transfer chain from photosystem 2. So again, we are starting at photosystem 2, and we're giving electrons from photosystem 2 to an electron transport chain. And then they take those to photosystem 1. So once the electrons are in photosystem 1, they are going to be ejected from photosystem 1 and go to, you guessed it, another electron transport chain. And then we're going to take NADP, and we're going to give him those electrons and that hydrogen that we got initially from breaking up that water, and we're going to get NADPH. He is very, very important for making glucose later on down the road. He's just as important, you could say, as ATP is. This guy's got to be built in the stroma. And so once we get ATP built, and once we get NADPH built, these are the energy products of light-dependent reactions in the non-cyclic pathway. So, energy products, useful products. Are there other little products in the system? Absolutely. But the two that you must know are ATP and NADPH. And we should probably throw free oxygen on there because he is so important to us, and we can be a little bit selfish there and make him sound more important than he is, because without him, we would die, and that would be just such a downer. So that's ATP and NADPH. Very, very, very important. Here's a not complicated cartoon. Said nobody ever. All right, let's take a look at this. Light energy, number 1. What a good place to start. Photosystem 2. In comes the water. It gives up some free hydrogens, which are great because we need those later. In come the electrons. The electrons are transferred to an electron transfer chain or transport chain, and they move the electrons, move the electrons, move the electrons. And while they're doing that, they're going to pull a couple extra hydrogens in. Those electrons make it to photosystem 2, which gets a little bit more light energy, and it re-energizes those electrons. and they go to another electron transfer chain, and they're transferring electrons and transferring electrons between proteins. If in case you were curious, if you don't know, guess protein. And we make NADPH. Hooray, we're halfway there. Okay, so we've got all these hydrogens in here, big old hydrogen gradient, and those hydrogens want to leave. That's called simple diffusion, and they're bouncing around looking for a place to leave, and they hit this guy, and he is a hydrogen gate that will let hydrogen go the direction it wants to go, down the concentration gradient from an area of more hydrogen to an area of less hydrogen. And when it does, it runs through this protein called ATP synthase. And when ATP synthase turns, he takes ADP, adenosine diphosphate, and inorganic phosphate and combines them to make adenosine triphosphate, which they are even considered enough to make a little coin because he's the energy currency, so we make him look like money. And now we have NADPH and ATP, and we are in really, really good shape. So that is half of photosynthesis. Just half. Yikes. Okay. Oh, wait, don't forget him. We like this guy. When water breaks up, oxygen leaves. Hooray. Waste. We like oxygen. All right. So that was non-cyclic. In case you didn't, non-cyclic. There is not a cycle. It is a straight-ish line with a definite beginning and a definite end, not a cycle. This guy's cyclic. And so a little piece of this is cyclic. When NADPH accumulates in the stroma, non-cyclic pathway is going to take a little bit of a break. So when we get too much of this guy out here and not enough of this guy, we'll actually stop this just for a little bit. So a cyclic pathway runs in our type 1 photosystems, photosystem 1, to make just ATP. So we're just going to run photosystem 1 and just make a bunch of hydrogens and keep that ATP synthase running. And it's going to make a lot of ATP. Electrons that are excess are going to be cycled back to photosystem 1 because we don't want to run photosystem 2 and keep making those NADPHs. And so we put a halt on that and we just make a whole bunch of ATP. And then we get a light-dependent reaction will continue even when our non-cyclic pathway stops. And so that's kind of a wordy way to saying if we need to, we can pause half of this light-dependent reaction to let our ATP catch up with our concentration of NADPH. So I know that seems like a lot, and that's because it is a lot. So here's the summary of your light-dependent reactions. If it's non-cyclic light-dependent, we can take ADP and inorganic phosphate and turn it into ATP. And we can still take NADP and turn it to NADPH. And we can take water and make oxygen. Now, the cyclic part of this is really just ADP and inorganic phosphate going to ATP. And so these guys work together to make sure that at the end of the day, we have enough ATP and we have enough NADPH to go ahead and make a lot of glucose. So that was light dependent. We don't want to start light independent yet because this is enough to kind of digest for now. I'm going to show you an animation that will summarize the points that we just discussed, and then we're going to call it good there. So hang on with me for just a quick second, and there it is, and we're up and running.
Photosystems consist of arrays of chlorophyll molecules. Chlorophyll, the green pigment of leaves, absorbs light energy. The absorbed energy excites electrons to a higher energy level. Energized electrons from photosystem 1 are passed down an electron transport chain and added to NADP+ to form NADPH. Meanwhile, energized electrons from photosystem 2 are passed through another electron.
Okay, so please note, I know this isn't like the most beautiful animation ever, but please note that he said meanwhile. It's not like photosystem 1 works and then pauses while photosystem 2 goes. They just break it into separate steps because it's easier for you and I to digest and learn. So just know that these two guys are running at the same time.
Transport chain. Their energy is used to pump hydrogen ions from the stroma into the thylakoid compartment, creating a concentration gradient. Electrons leaving this electron transport chain enter photosystem 1, replenishing its lost electrons. Photosystem 2 replenishes its electrons by splitting water. hydrogen ions and oxygen are released into the thylakoid compartment. This is where the oxygen gas generated by photosynthesis comes from. The buildup of hydrogen ions inside the thylakoid compartment stores potential energy. This energy is harvested by an enzyme called ATP synthase. As hydrogen ions diffuse through ATP synthase down their concentration gradient, the enzyme uses the energy of the moving ions to make ATP. Next, ATP and NADPH are used in the sugar-making process of the Calvin cycle.
All right, so hopefully that was helpful. Cancel. I don't want to go on to the next video. Sorry about that. But hopefully that was helpful for summarizing how we get and where we get our reactants to convert into our products, and also where those reactions are occurring and where our products are released to, because of course all of these end up in the stroma. So light-dependent reactions, it's a lot to kind of take in, but I hope it was helpful. And if you have any questions, please don't hesitate to ask me. So thanks for your time, guys.